People are freaking out for this one. So I will talk about that now. Obviously, this paper is a bit different in the sense that you don't have introduction written as introduction, methods written as methods, results written as results. It's a bit more like a review because each section has its own title, but it is still an original research paper, okay? The other thing that this paper has that is kind of weird compared to the other one is the fact that it's referring to previous work that the team has done. And so where do you stop, right? I was waiting for people to read it and then start asking me questions because I wanted to see if you were able to do this or not. So let's do it. No. Yeah, this one. Okay, so this is the paper I assigned to you. So obviously, if you go through it like this and you read, you'll see that there's nowhere where it mentions methods except at the end, which is like maybe 6-10 lines. The thing is, the actual methods are in the supplementary information, which I guess you skipped. Okay, so if you go to the supplementary information, you will find everything about the methods, from the sampling to the DNA extraction to how they prepared the libraries to how they made sure that the sample was not contaminated, and their computational analysis, their error rates, the whole thing, okay? So there's this here, and so if you read this part here, you can figure out a lot of the things related to the methods. So that's the first thing. Obviously, when I'm going to talk about it, I'm going to use these PowerPoint slides, okay, and add a little bit of information for myself. The other thing is, you kind of have a purpose, right? They're saying what they did, and what they tried to do, and what they achieved, and what they don't know. Okay, so a known archaic gene flow in, right? So they don't know this. You know that their goal was to kind of figure out the gene transfer between different groups of hominids or hominins, and then they tell you what they did and how they analyze it. It's not in the test tube, right? They're not, yeah, of course they extract the DNA, they try to make sure that it's clean DNA and all that, but it's not like the other paper where they're doing like screens, knocking down stuff, looking for phenotypes and things like this. Here it's more like comparing things between them. Right? So there's still a flow from a goal to an endpoint, and they do things to get there, right? Now, in terms of discussion, obviously their discussion is a little bit smaller than the other paper. So that means you need to be a little bit more creative. Where the hell is it? Anyways, what is this thing here? Just 1 second, guys. Here, clicked on the wrong thing. So it's kind of tiny, right? So obviously if you're using the other paper, in the other paper, like their discussion and then building on it and paraphrasing somehow, now it's going to be a little bit harder to do because their own discussion is kind of smaller, right? But you can also just expand on what the hell, why is this interesting? Why the hell do we care about this stuff, right? And has this been used, for example, to achieve anything related to evolution or to understand diseases, or I don't know, right? So you can expand on this stuff by looking at more than just what's written in here, right? So that's okay. It's an open assignment, so obviously some people will just focus on the paper itself. Others will expand here and there. There's no real format other than summarize the paper and tell me what's wrong with it, what's good with it, and what's the future, let's say. Obviously, if you go in the methods section, you have a summary that is here, but you have a supplemental information that you can figure out how they did all this stuff. You can discuss the contamination rate, for example. This is a thing. I was trying to avoid doing this so that I don't give you all the answers, but I guess it's a good teaching experience, kind of. So you can talk about the contamination, right? Because this is a sample that has been preserved from a long time ago. So what tells me that DNA is not shit, right? maybe it's really bad DNA and we're just hallucinating and inventing stuff, right, about that DNA, because you can extract the DNA and you can say it is that, but there's a rate of error that comes with that. It's not perfect. In the lab, when we extract DNA, we try to keep it in on ice, and there's a reason for that. Now imagine something that comes from thousands of years ago, right? So obviously this bone, if I remember correctly, was found in Antarctica or something like this. So it has been preserved, but it's not in the perfect laboratory conditions, and they do things to make sure that this is good, right? Obviously there is a rate of error that is intrinsic to this kind of study, and so you can talk about that, for example, in the discussion. You can talk about the decision for how they decided to assign nucleotides or whatever, right? So just read it, take 2-3 main points, talk about those 3 points. If you run out and you run out of information to say about those 3 points, add a 4th one and just keep going, okay? So choose whichever ones are interesting to you. It's open-ended, okay? Does this make sense? Good. Any questions? Yeah. Same as the first one, 4 to 5. Yeah. There's data. Talk about the data. Yeah, I understand. It's a bit weird when you get something like this and then suddenly the professor switches it a little bit. But it's a good exercise so that you have an idea about the different papers that come out. What else? I think that's that. Okay. So we're almost done. Soon you're going to get rid of me. That's it. So we have one last topic to cover, and then I'll be talking about the 2 papers and then some other interesting papers. So today we'll be talking about functional RNAs, which are quite interesting, and there's tons of them, and we keep discovering new ones every year or so. And so this is the last thing that is part of the genome structure. After being sequenced, you have all these pieces that we know about. If you one day discover another piece, then it will be added in my slides right here, okay? But for now, this is the ones that we have. And functional RNAs are quite interesting. They're so interesting that there's many Nobel Prizes that were won related to the functional RNAs because they have major roles. 2% of the genome, or not 2%. I forgot the percentage, but basically a very small percentage of the genome is protein coding. The rest is mostly functional RNAs, transposons also. And so obviously they have a big role, and they're linked to many things like disease and such. So many people made a lot of discoveries in these fields. So what I'm going to do is go through the different categories of RNAs and talk about a little bit the important people that happened in this field. And because if I get too much into the structure of each one of them, it's way too much, okay? We could do it in like half a semester. So I'll just give kind of the structure of 1 or 2, maybe 3, and then the rest is just tell you what it does, an example, and that's it, okay? So all this stuff here came from, let's go back a billion years ago. Anybody know the RNA world evolution theory? Do you remember that from 225 or something like that? So basically, it says that we come from RNA, and some volcanic explosions happened and molecules mixed up together, and suddenly they gave rise to RNAs, okay? So we think of proteins as being the enzymes that do kind of the function, and then RNA and DNA to carry the message, right? But in this theory here of evolution, we think of RNA as being the one that's able to do both the coding and the catalytic functions. And as a matter of fact, we thought for a long time that this is wrong, but there was a Nobel Prize that was awarded to a person that showed that these are catalytically active. So this could be a potential thing, but obviously we will never know, or at least not anytime soon. What's important though is that you'll see that this makes sense because there's many categories of non-coding RNA that do different things. And so there's a lot of data that supports this theory. It's not just some random, some guy was sitting in his lab and thinking about this stuff, right? There's stuff that supports this. Obviously, it seems weird that some volcanic explosions made some molecules and the molecules started replicating. Then they acquired functions, but there's a possibility, right? And so in this theory, the model says that the proteins came later to actually stabilize and improve the function of these nucleic acids, but they're not pretty much required. Okay. There's all these. These are all the categories of functional RNAs. There is the ribosomal RNAs, which I'll talk about today, tRNA, transfer RNA, small nuclear, small nucleolar, mRNA or microRNAs. We'll talk next lecture about the rest, okay? So that's that. So just, I think you've seen them all and you keep seeing them all, so I'll try to make it quick, not boring. This is why I did a little bit about the Nobel Prize, because I know that you've seen before Drosha and Dicer and antisense and sense, so I'll try to kind of avoid that, okay? So Nobel Prizes, there's I think 7 of them or 6 of them. So I'll tell you what are the main players. So the first Nobel Prize that was linked to RNA was in 1959. That's from Ochoa and Kornberg, which basically discovered the biological, not chemical, biological synthesis of RNA and DNA. So this is where everything kind of started for this molecule. Then there's these 3 dudes, Nirenberg, Hawley, and Horna, I don't know. That got the prize in 1968, and they basically wait 1 second. They basically figured out the code. Jeez, this is hiding my text. And 1 second. Okay, what? Okay, I don't know why this. I'll just hide it here. Okay, so the 3 unit codon was from them. They understood how DNA brings mRNA, and then how mRNA brings proteins, kind of. And so this is very important, but mostly theoretical. The cool stuff starts a little bit later when Mr. Altman and Czech, I believe, got the Nobel Prize in 1989 for their discovery on the catalytic function of RNAs. So these are the 2 guys that showed that maybe the RNA, brought information that showed that maybe the RNA world theory actually is true. So we'll see today that a lot of the RNA molecules do have a function. They're not just there to carry information. And so this is the first time that was kind of shown. Then in 1993, the year I was born in, these 2 guys, Roberts and Sharp, discovered the split genes. So they saw that actually a gene was split into smaller pieces, and they had some sort of DNA molecule or piece of DNA that was in between, and then you don't find that in the mRNA because of splicing. Okay, so they used to call it split genes, but now they realize that it's actually the same gene, just spliced differently. There's a very important group of molecules that act in RNA splicing, functional RNAs, called the small nucleolar RNAs. Small nuclear RNAs. And we'll be talking about that today. 2006, maybe a few of you remember this. This is where RNAi was kind of discovered. It was discovered by Andrew Fire and Craig Mello. And this is a very, very important discovery, because this is one of the ways that we use in research, actually the main way currently that we use to regulate gene expression. And so if we're trying to study it in terms of diseases and things like this, we'll use RNAi to do that. And there's a few types: microRNA, siRNA, shRNA. So these guys killed it, basically. 2020, you guys know these 2. You've probably seen it in the news, CRISPR-Cas9. So Emmanuel Charpentier and Jennifer Doudna, they developed CRISPR-Cas9. Now there was a little bit of controversy about this stuff here. that maybe someone else had it before, but in any case, it doesn't really matter. They got the Nobel Prize. So they built the CRISPR-Cas9 technology using bacterial CRISPR system, which is kind of their immune system. And this involves single guide RNA, another type of functional RNA. 2023, COVID. You guys remember these? So before, we used to make vaccines by using kind of proteins from the virus or the infecting agent, and then, or we actually take the viral particle and then just make it a little bit weaker and then put it inside. But now, one thing that was preventing us from using mRNA, which is a lot less dangerous, let's say, although not risky, but let's not get into that. The mRNA, the problem is that when you take mRNA that is synthesized in the lab and you put it in your body, the body recognizes it as something foreign. And because it is foreign, there's an immune response that happens to you, okay? So we've learned RNA as being like this very simple molecule that has only A, U, C, and G. But actually, it's not that simple, okay? If you look more carefully, you'll see that there are other nucleotides that exist which tell your body that this is your mRNA or an mRNA from outside. And so when we used to try to make these mRNA vaccines, you would get a kind of a reaction in the body of the mice or whatever you're testing on because they recognize it as a foreign agent, and that would just kill the mice, right? So these guys found that if you modified the mRNA before putting it into the cells, using multiple modifications that we'll talk about today that are done by the small nucleolar RNAs, you will then kind of evade this immune response, okay? One of the modifications is called pseudo-uridinylation, whatever. You change the uridine to a pseudo-uridine, okay? And this is a SNO RNA that does it, and that will tell basically the body that this is your own RNA. So their discoveries made the mRNA vaccine possible, and this is basically the basis of all the vaccines that are used for COVID that are using mRNA technology, okay? The last 2024, there's a mistake here. This year, there was a Nobel Prize won by Gary Ruvkun and Victor Ambros. I talked about them quickly, but these guys discovered microRNA and their role in post-transcriptional gene regulation. So genes are regulated by miRNA in your body. siRNA and shRNA are less regulatory. It's more miRNAs that are kind of the natural gene regulation in cells, okay? You'll see that thousands of diseases are related to miRNAs. So I'll talk about this a little bit today so you can see how important this is. Okay, let's start by a small little question. This is a ribosome, obviously. I have a question for you. What do you think is the biggest component here? Protein, RNA, or they're the same? I mean, obviously it's RNA class, so. Okay, so the ribosome has more RNA than protein in it, okay? So you see all these little wiggly stuff here in white and in black. These are RNA, the rest is actually protein. And there's two types of RNAs that exist in the ribosome, the ribosomal RNA and the tRNA, or these guys. So the tRNA is one of the type of functional RNAs that exist in the body. Sorry, you'll hear this word RNA all lecture long, but can't do any other way, okay? So the tRNA has a specific structure. It looks like a clover leaf like this, and it has these 4 loops. So 1, 2, 3, and 4. And its role is very simple. It's to basically assign an amino acid to a specific codon. So when you're building your protein, these tRNAs will be bringing the amino acid to the ribosome, and then we'll be adding it to the polypeptide chain. There's a few features that are important for its structure, and I'll tell you about that in a second. But basically, you have the anticodon here. You have the carboxy terminus here, which has a specific CCA tag, which is the attachment site for the amino acid. And these loops here that are just important for placing the tRNA inside the ribosome. Okay, so this is what it looks like. I know you've seen this before. The ribosome has two subunits, the large and small subunit, and these are made up of multiple proteins and RNAs. And there are a few sites within the ribosome where you have the transcript passing through, and you have the polypeptide being elongated because of the addition of amino acids that are brought by this tRNA. So the tRNA will enter here, will leave from here, and while it's passing through, it will add to the polypeptide chain. Okay, it enters by the A and then leaves by the E. Okay, and every time it will come here, it will use its anticodon, which is the opposite of the codon from the mRNA, and recognize it here, and then bring the amino acid here. Okay. What else? Few features that are important. So in the tRNA, there is the sequence on the carboxy terminus of the tRNA, which is CCA. This is where the amino acid binds. And so if you look at the structure of the tRNA, you'll see that it needs some sort of specific sequences of nucleotides to have its function. Okay. First, you need nucleotides that interact together to form these loops. Second, you need a specific anticodon. So there's many tRNAs that exist. Each one brings a certain amino acid. Some of them will have multiple tRNAs that can have different anticodons bring the same amino acid. So you will need this piece of anticodon here, and you'll also need in your structure the amino acid attachment site. So if you are looking for all the tRNAs in your genome, then you can look for these features. Okay. Usually these RNAs have about 75 bases long. Obviously this diagram is not true, it's a bit bigger than that. But if you're using computational methods to actually identify all the tRNAs, let's say in a new organism, you'll probably start by looking for something that is 75 bases long, has an amino acid attachment site, has an anticodon at some sort of position, and has these features here that allow it to form these loops. Simple? Easy? Okay, good. One important thing is the tRNAs are modified. So this is what I was talking to you about. You have to modify your molecule, your RNA molecule, so that you prevent them from creating kind of this immune response, because your system will detect them as foreign RNA. So in your messenger RNA, you have the cap, right? And the cap is one way to protect your mRNA from causing an immune response. But there are other ways. For example, the tRNAs, when they are formed, they are then altered by enzymes that can basically change the uridine to dihydrouridine or to pseudouridine, and also inosine. Okay, these are just different types of nucleotides, and these tell your body that this tRNA is yours. All the tRNAs, almost all, they have this inverted L-shaped clover leaf formation. Okay. Cool. So another structural feature that is important for tRNAs is the fact that they have these nucleotides on the side close to the antisense codon that allow it to not react, but to kind of, interact with the amino acyl tRNA synthetase. And this is just the enzyme that will basically make the tRNA mature, helping the maturation of the tRNA by adding the specific amino acid to it. Okay? And then this enzyme here is just adding amino acids to each of the specific tRNAs. Now, this here is very interesting, because unrelated to the course, but I'll just tell you what's happening around there. You have a protein that is able to release a factor based on some sort of compound that is bound here, right? So you could modify this protein here to detect a toxin and then release a drug, right? And this is happening right now. There's a lot of research that's happening on this. There's a lab that I worked in that worked on tRNA. Paul Joyce, they're not doing this kind of work. This is bioengineering, but they're working on the link between tRNA mutations and diseases. But let's say, for example, you do an RNA-seq of a cancer patient, and then you have the healthy patient, and you compare their RNA set, and you realize, or proteomics, whatever, and you realize that they have this kind of metabolite that is in high levels, right? So that means the cancer makes that metabolite go up. So then you can use that metabolite and use this protein here to detect it and then release some sort of chemotherapeutic drug, right? So there's a lot of work that is being done on this. Seems a little bit simple. It's not that simple. But if you take the sequence of this protein here, you have an amino acid binding site. So this is a domain, right, that has a motif that allows for the binding of the amino acids. So then you could change this, you can change the sequence to allow for binding of something else. For example, if you have lactate that increases at super high levels because of cancer, there are proteins that sense lactate in the body. So then you could take the lactate sensing domain from another protein, put it right here, and then change this to hold actually, you don't even need to change this. Leave the tRNA and then conjugate it to a chemotherapeutic drug, right? Crazy stuff. There's a lot to do. If only I could be immortal, I would fix all this stuff. But, sometimes you got to choose something and go with it. So choose wisely. One last thing about the tRNAs before I move to the next topic is the anticodon. So these anticodons are quite important because they will recognize they're basically the link between the RNA codon and the amino acid being added. So most of the diseases caused by the tRNAs are usually in the anticodon, and either in the anticodon or in the loop formation structures. And so obviously if you have a tRNA that binds to a specific amino acid, but for some reason it's using the wrong anticodon, then you'll end up carrying the wrong thing and binding to the wrong thing. So this is a big problem. But forget about the wobble position. Just useless for this class, but it just means that the last nucleotide usually does not matter for determining which amino acid is linked to this codon, okay? 2-second breathing break. Okay, so ribosomal RNAs. Let's look at that. So as I told you, the ribosome has a lot of protein, but also a lot of RNA. They have these ribosomal RNAs and these tRNAs that come in and out of it. The ribosomal RNAs have a different structure, this weird shape here. They're a little bit longer than the tRNAs. And they have one very important part, which is usually the sequence that allows it to bind to the mRNA. Okay? So the role of the ribosomal RNAs is to actually link the ribosome to the mRNA, to hold everything together. And they do this by having a specific sequence that recognizes another sequence on the mRNA transcript. Okay? So for bacteria, for prokaryotes, the sequence is called the Shine-Dalgarno sequence, and it's found about 8 bases before the start codon. So you have your transcript. How do you place your transcript within the ribosome? You need some sort of way to align it, right? Because you want the E, the P, and the A site to be perfectly aligned. And so the way you do this is through the ribosomal RNA. Because the ribosomal RNA has a sequence that is used as a target recognition sequence, which means it will find the opposite sequence and then bind to it. And this ribosomal RNA is also stuck on the ribosome. In eukaryotes, this piece of sequence is called the Kozak sequence, and there's many different variations. Okay. What else? Obviously, different ribosomal RNAs, they recognize different sequences, but you can see how RNA is used a lot by the body, by the cells, as a kind of target recognition system, right? In the tRNA, they recognize the codon and bring the amino acid. In the ribosomal RNA, they recognize a certain sequence to align the ribosome And you'll see in the others, they also have this kind of target recognition system, okay? Small nuclear RNAs. So this is another group of functional RNAs. These ones here, they are very important for splicing, the 1993 discovery, so based on this stuff here. So the small nuclear RNAs are found in the nucleus, obviously, and they will bind to proteins and create these small nuclear ribonucleoprotein particles. There is 5 of them. So small nuclear ribo blah, 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 blah is basically SNRPs, okay? So we call it SNRPs. So there's 5 SNRPs, U1 to U6, and there's the U. U is used just to remind us that it's a sequence rich in uridine. So this is one of the features of these small nuclear RNAs. They're rich in uridine, okay? They will combine all together, and they will form the spliceosome. So each U1 will recognize the splice site. So the first splice site, so it's basically just the small nuclear RNA is basically the opposite of the splice site. And obviously it's bound to a protein, these ribonucleoproteins. And so they will all interact together and flip your RNA, your pre-mRNA into kind of this loop here. And then they also have catalytic functions. So these SNRPs, they have a catalytic function where they can actually break the RNA and then bring it back together. You don't need to know the specifics about this. Just know that these small nuclear RNAs are used in this case to recognize the splice donor site and acceptor site and to flip the mRNA, cut it, stick it back together, okay? When they cut it, they form this intron lariat, which is this kind of circular intron piece that is then just degraded, okay? Questions? So small nuclear RNAs, their goal is to recognize the splice site and also to bind to these ribonucleoproteins to do all these functions that I just told you about. Small nucleolar RNAs. So remember I told you about the pseudouridylation. So these SNO RNA family this family of functional RNAs is important in modifying other RNA molecules. So this is their only goal. What they do is that they will go grab the RNA molecule, modify it, and that's it. And usually they will modify the tRNA, the ribosomal RNA, and there are a few, 2 types of them. One of them is the, I don't know why they put these weird names, but HACA type, which turns a uridine into pseudouridine. And this is important, as I told you, for recognizing the RNA as your own, okay? The human telomerase RNA has this motif. The mRNA vaccine has used this technology to build the mRNA vaccine. And then there's another type which created kind of this new field. So you've heard epigenetics, right? About epigenetics. Well, there's also epitranscriptomics, right? So it's just same thing as epigenomics, but epitranscriptomics. So it's looking at the modifications of nucleotides in RNA molecules. So the CD type of SNO RNAs are used to do, for example, methylation. And they do methylation on certain pieces of certain nucleotides. What does this do? Change the form of the mRNA, prevent it from being degraded, things like this. As you can see, these very book-like drawings. It's very hard to find nice illustrations for these guys, probably because they're not that well studied, but there's that. Not bad. Almost done, guys. Quickie. So, microRNAs. This is the cool stuff. Anybody heard about this Lin4, Lin14 stuff? Okay, so you keep hearing about it until 2024. They got the Nobel Prize for it. I still remember my classes from genetics that talked about this, advanced genetics. So this was done in C. elegans. There is an miRNA molecule that targets a gene, right, called the Lin14 gene. And so these microRNAs are very important in your body because they regulate the amount of a transcript in your body. So not just gene or protein producing transcripts, but any transcript. And usually the sequence that is required to recognize that same transcript is found in the transcript itself, okay? And I'll show you that in a second. But these microRNAs, they form a hairpin loop like this, which resembles all the loops that we've seen before, but it's a little bit different. It looks like the short hairpin loop found in shRNA, which is SH. So this is RNA interference, and they look similar. They have similar mechanisms. They use the same proteins. It's just they do their silencing in different ways, okay? So the miRNA binds to a protein called dicer and gets prepared into this form. And obviously, the sequence here, what it does is it recognizes the opposite, the complementary sequence on a transcript, right? It binds to it, brings a protein with it, the RISC complex, and that RISC complex will break the transcript. By doing that, you prevent the transcript from being translated, and you decrease the expression of whatever protein, okay? So in this case, for example, the Lin4 and Lin14 group, which were studied in the C. elegans. So these two were mutated, and when you mutated one of them, you saw that the shape of the C. elegans kind of changed, right? It's not shown here because I know you guys already seen it. And so it kind of changes the muscle shape of the C. elegans. And because it did that, then they found that one of them was regulating the other one, although they were not cloned at all, okay? So just by mutating these two, they saw that they could reverse the effect of the other. And so this Lin4 mutant, this Lin4 miRNA is a microRNA that is found in the transcript of the Lin14. That is found in the Lin14 transcript. So basically, you have the transcript. The transcript starts here, ends here. You have your poly A tail. You have the ORF, which produces your protein, right, for the muscle, whatever. And then at the end of it, you have these sequences that can form the microRNA. So they're already encoded in the transcript itself. And obviously, it makes sense, right? Because if you want to silence a transcript, what's better than using the transcript itself as a target recognition sequence, right? So there's a few of them that are found at the end of the Lin14 transcript, and they all kind of can silence the Lin14 expression depending on which one is being used. They also have another effect. So whenever these microRNAs are bound into the RISC complex, they also have a direct inhibitory role on the ribosome. So they do cut the Lin14, break it down, but they also inhibit the current ribosomes that are bound to it. So there's a few ways in which they can actually stop the transcription of this, translation of this mRNA molecule. In terms of structure, I'm not getting too much into it because there's other pieces here that are not shown that get broken. But if you're looking for this inside your genome, you'll probably be trying to find sequence that is following each other and that can form this kind of structure, right? So you need kind of the opposing nucleotides that are in a sequence that can then form this shape here. Okay, and you can use just the computer to do that. It's not that complicated. Easy? Kind of? Yeah. It's pretty repetitive. We don't really get too much into the details because there's way too many categories and way too many specificities for each one of them. So just go through them quickly. So these miRNAs, they're first transcribed in the nucleus. I know you've seen this, but we're going to go over it again one last time. They're transcribed in the nucleus, and they get processed by a complex called DROSHA and DGCR8. It's the name of a disease. I don't remember it by heart, but basically they will cut the first piece here to prepare the primary microRNA, turn it into cursor microRNA, which is then exported outside. When it's exported outside, it will be prepared to be into a single strand of RNA molecule that then will be used to recognize the transcript. Okay. The reason for this shape here is to avoid its degradation until it can reach this stage here. Okay. Because if you leave this hanging on in the cytosol without any protein bound to it, it will be degraded. The complex that binds this is called the RISC complex, which does also the cutting of the transcript and the inhibition of the ribosome. Okay. RNAs are often studied in the same kind of thinking as transcription factors, because if you think about it, really, transcription factors, their role is to actually induce transcription, and these guys is to prevent translation. And so, although this is not 100% true, but transcription factors sometimes have an inhibitory role also. The transcription factor itself stimulates transcription, but usually they interact with other cofactors to do the opposite effect. But in general, their goal is to promote transcription, right? While miRNA have the opposite effect, their goal is to prevent that protein from being produced. So whenever these guys are active, these guys will try to shut down the effect of the transcription factors. So this is kind of the control that goes between these two. And obviously, transcription factors will activate the transcription of RNAs specific for the same transcripts that are being produced here, because you want to make the level of those transcripts, regulate the level of those transcripts. Does that make sense? So you're trying to make proteins, so you make the transcripts, but you also have to make the microRNAs that will kind of reduce that at some point in the future, okay? Because you don't want proteins to be active nonstop forever. The transcription factor obviously are control the genes at a transcriptional level, while the microRNAs at the post-transcriptional level. And here's a few examples here for different types of RNAs. So microRNAs, as I told you, are found within the transcript itself that they're trying to silence. So MIR21, I forgot what disease this is linked to, but I think MIR21 is like cancer, okay? You can probably Google it, but the microRNA that is produced from this transcript is actually targeting, is found on it, okay? And there's a few different ones, so depending on which transcript you're working with, sometimes you'll have multiple ones because you need a stronger regulation of that transcript, other times only one. So depending on the level of regulation that you need. You also have non-coding RNAs. So although non-coding RNAs, they don't make proteins, you can't keep them always there because they have a functional role. And so you want to also silence those. So also those have a microRNA. One interesting thing about these is that you'll always find them at the end of the chain of your RNA molecule, right? So you'll have, for example, in this case, these mRNAs. These mRNAs will produce a protein. So you have your ORF, because your close to your cap is your start codon. You'll make your protein, and at the end here, you have your microRNA. It would be weird to have it at the beginning and then have the translation happen from here. So this is just kind of makes sense, okay? They're found in introns also. This is very important for the gene producing, the transcript, the protein producing genes, okay? 3' UTR, I guess I can explain this. So the 3' UTR is so the mRNA molecule has two regions, the 5' UTR and the 3' UTR. These are untranslated regions, so the ribosome does not start there. It starts at the start codon, so there are regions that remain on each side that are not transcribed. These are very important for gene silencing because if you're trying to do, for example, gene therapy, you want to target the messed up transcript in the cell. And the way you do that is by targeting the 3' UTR of that transcript, because then you'll try to put a transcript that is protein coding inside the cells, and that transcript will not have the 3' UTR. because it's basically mRNA converted back to cDNA. Does that make sense? So then you'll use this mechanism so that you avoid degrading the mRNA molecule that you are putting from your, like, to fix the problem. So let's say, for example, cystic fibrosis, favorite example. It produces an mRNA molecule, and that mRNA molecule is messed up. So you'll use some sort of RNAi that targets the 3' UTR of the messed up transcript. It will get it degraded, and then you'll put back in the cells the good transcript. And that good transcript, because it was made from mRNA and then back to cDNA, because you put in the cells mostly cDNA, then it does not have the 3' UTR. Okay? And so this way you can avoid degrading the nucleic acid that is going to save you. Okay? So this is a high therapeutic potential here. And for some reason, these microRNAs are always found there. Almost done. 2 slides. Easy week, so you can go party. So the mechanisms by which these microRNAs actually prevent transcription, translation is through 3 things. So when you have an active translation that is happening, you'll have your initiation factors, proteins that recognize the cap and the poly-A tail and then allow for the ribosomes to bind and do their job, and the ribosomes are just transcribing. If you have this microRNA, what will happen is that your microRNA will bind to a RISC complex, and that RISC complex, one of the proteins called Argonaute, we don't care, just to tell you what's happening, will come and recognize some sort of sequence at the end, right here, and it will kick out all this stuff here. When it kicks out the eukaryotic initiation factors, which are important for translation, then they cannot recruit more ribosomes. In addition, what they will do is that they will directly inhibit the ribosomes that are already bound to get them off from the mRNA molecule, and they will also promote the degradation of the mRNA molecule to decrease the translation of that transcript. Does that make sense? In terms of structure, very quickly, you need a molecule that is able to bind specific proteins and recognize a sequence. So it's always like this. It's just they're used in different purposes. Okay. So the functional RNAs are always recognizing some sort of sequence, and they're always they're always bound to some sort of protein or amino acid. Okay, because they need some sort of enzyme to do all this work or recruit other proteins that are important. Last question. How can you find all the mRNAs in the genome? Yep.
We can do high-throughput sequences like RNA-seq.
Yep. But how do you know it's a microRNA? Yes, Alessandra.
Using the hairpin structure, so we can look for, let's say, complementary nucleotides, and then just expand from there.
Right. Is this easy to imagine? Kind of? No? So you have your gene on your DNA, okay? It has a sequence. So when you see a sequence, and then that same sequence is repeated here, but kind of complementary to this one, when you make the mRNA, most probably they will bind to each other, right? And so there are ways to figure out if you will be having this complementarity happening when you make your RNA molecule. Now the thing is, all RNA molecules can be complementary, like can have complementarity. Even if you're making a primer for some sort of homework, you can have complementarity, okay? And so how then do you know if it's a microRNA or a messed up primer? Yeah?
Maybe you can see with the transcription factors level if it's similar, because you said we can study the effect pairwise.
Yeah, so you can increase the expression of these microRNAs by promoting transcription of certain stuff, and you'll see that some stuff, like for example, if you do, if you do ChIP-seq, you can see that your transcription factor is binding at many places, and those places potentially have microRNAs. And then you have to look at each place where your transcription factor binds and look for that sequence that I just told you about. Yep.
I was going to say you do the same sort of gypsy thing, but with the RISC complex and all the mRNA.
That's it. I like this. That's a very good answer. So you can actually cross-link the protein to the RNA molecule. So if you're studying microRNAs, you can actually freeze the protein on the microRNA, so RISC complex, do an immunoprecipitation to bring that RISC complex, break the protein, sequence. Make sense? Very good answer. That's I like it. This is research level stuff. Okay, so there are a few ways to do this. Obviously, yep.
Can we use comparative genomics?
Sorry?
Comparative genomics.
Yeah, you can also do that. So if you find it in one organism, you can go backwards.
So those are microRNA.
Yeah, the LIN4, I'm not sure if it's found in humans. Does anyone know? No? Probably. But yeah, you can look, you can compare different organisms between them and see if they have the same microRNA. So there are a lot of ways to do this. We've learned a little bit about how to sequence the genome. Now it's how to find all the pieces, right? So you have to be a little bit creative. Obviously, the final exam, I'll probably ask you something like this, but you don't need to have like the perfect answer of I'm going to do a chip using the RISC complex, right? If this does not come instinctively, it's normal, okay? You're not expected to actually think about this, but you have the tools that I taught you in this class and probably in functional genomics if you're taking it too, to actually do this thinking and think about it. And usually the way you think about it is written in the title, okay? Structure, right? So there are specific structures for each piece. If you're looking for a microRNA, it has a specific structure, right? Now, I didn't talk about it a lot. All I said was it has a hairpin loop that looks like this, which is kind of similar to short hairpin RNA, but a little bit different. I talked also about the complementarity. So that's your way of thinking. Okay, so you're looking for this kind of feature inside the genome. All right. With that, if you don't have any questions, that's it.